Okay, so just a little bit more about the lac operon. I'm not sure we talked about this part, but early on in chapter 2, maybe, I think it was chapter 2 or 3, we talked about molecules outside the cell. When they're in abundance and they get translocated into the cell and they immediately get changed into something else, it helps with that gradient, because if you look at this, on the outside of the cell, there's lots and lots of glucose, and on the inside of the cell, there's no glucose. As soon as the glucose gets pulled in, it gets phosphorylated and it's no longer glucose, and that keeps a steep gradient. Lots on the outside, none on the inside. It helps to move that glucose down inside, because as soon as it gets in, it's being phosphorylated to that famous chemical that you know at the top of glycolysis called glucose 6-phosphate. It's immediately changed to that. There's no free glucose in the cell, and then it helps to bring more glucose in. If the cell starts to run out of glucose, then some other things happen, and that other processes start to happen. And then if we've got lactose in there, there's something else that goes on in blocking the permease, and then other things begin to happen. And now cyclic AMP level goes up, and you see that being indicated here. The cap activator protein combined, it actually then recruits RNA polymerase to come in and bind to this promoter. In other words, RNA polymerase can't bind to this promoter by itself. We've talked about other scenarios where RNA polymerase can't bind, and therefore it can't start transcription. Some of those that we talked about involve sigma factors, remember? And alternative sigma factors are produced. They recruit the RNA polymerase to those other genes. That's the case for sporogenesis and a number of other things. That's what's happening here too, except for it's not a sigma factor. It's this other activator protein that binds here, and you have to have that co-activator, the cyclic AMP, being high enough concentration to get it to be able to bind. Once the cap protein binds, RNA polymerase is recruited in, and then you get transcription happening. But like we just saw, if lactose is used up, the cell doesn't want to invest any more energy in making that messenger RNA. So as soon as lactose is used up, what's going to happen in that scenario that I just highlighted? The repressor protein will jump right here, and it will block the RNA polymerase. So at the top of that dioxide growth curve, we were seeing this scenario. Cyclic AMP is high, and so the activator protein's bound. That's going to recruit RNA polymerase in, but RNA polymerase can't do its job because this repressor protein is bound here. But as soon as we throw more lactose in, then we're going to have allolactose produced, right? And then the repressor protein will be dropped off. It won't be there anymore, and then transcription can happen. Everybody's good with that, right? Cool, cool, cool. All right. So when we think about eukaryotes, many times regulation is more complicated, and that's because of a number of reasons. Some of them we've already discussed. In prokaryotes, we've got those organized genes that are all transcribed together that we call blank. What are those genes? That makes a polycistronic message, and on the chromosome we call them operons. Operons. They're one group of genes, open reading frames, that are all transcribed together to make a polycistronic message, and on the chromosome they're called operons. And if we have two or more operons on the chromosome in a prokaryote, then we call it a regulon. Remember that? And so in eukaryotic cells, we've got a little bit different situation. We don't have polycistronic message typically. We'll have one gene on chromosome 1, another one on chromosome 7, another one on 15, and maybe another one on chromosome 20, and another one on 21, and they all need to be turned on at the same time. And so the mechanism is a little bit more complex, but it works the same way. You've got these activator proteins or these repressor proteins that bind individually to each one of those promoters, and they all get the same signal down inside. Another way that transcription is controlled in eukaryotic cells is sometimes the chromosome structure is modified. Sometimes we get the nucleotides actually being modified just a little bit, and now we know that methylation on the chromosome actually does a lot to control the expression of the promoter. And again, as you think of those proteins that are doing all this processes, they are enzymes. And remember, enzymes only recognize certain substrates, and as soon as you change that substrate and add a methyl group onto those bases, it's not the same substrate anymore, and the enzyme then can't bind as well, and it can't do its job as well. So that's one way that the processes are modified in chromosome structure. The regulation of transcription happens in a lot of ways similar to what we just talked about, but many times on a eukaryotic chromosome, if you've got the gene here, and this is the open reading frame over here on the DNA, I should put ATG on the DNA and a stop codon over here, and then you've got a promoter sequence over here. I'll just put a P above it so you remember that's it. In eukaryotic cells, those activator binding sites many times are very far away from that promoter. Sometimes there are a few thousand kilobase pairs or a few thousand base pairs away from the promoter region, but sometimes they're 20 or 30,000 base pairs away from the promoter region, a long way away, and somehow the proteins bind over here, and after they bind here, they influence what's happening way over here in the chromosome. So things are much more complex in the eukaryotic situation than they are in the prokaryotic situation. Then we got to think about another scenario. You're a cell that's clicking along, you're making messenger RNA because life's good, you've got lots of nutrients, you want to turn on that messenger RNA, but now let's say you're E. coli and you made a whole bunch of RNA to make those 3 proteins that are needed to use lactose. You did it because there was a lot of lactose in the environment, but now you've used the lactose and there isn't any more lactose in the environment, so you shut down the promoter, you don't make any more of those transcripts, but you've still got a lot of transcripts that you already made that can be used to make those proteins. But because there's no lactose around, making those proteins is a waste of energy. Remember from chapter 6, every amino acid that goes into a protein could actually be used to feed into the TCA cycle to make a whole bunch of ATP in that TCA cycle if you have one. And so when you pump them out to make those proteins, you're actually wasting energy because they could have gone on through and you could have gotten lots of ATP out of the TCA cycle and the electron transport chain. But now you're building proteins. If you don't need those proteins, you want to stop. So how do you stop? You got the messenger RNA, it's already made. As soon as the ribosomes jump on it, they're going to do their job. They're going to start doing translation on that messenger RNA. So that one way to shut down that process is to get rid of the messenger RNA that's already been produced. Obviously, you already blocked transcription, so you're not getting more messenger RNA. That's what we just talked about. But we've got a whole bunch of messenger RNA that's now useless to the cell. If they make those proteins, they waste energy. So there's a system that's called RNA interference, and we've really just begun to understand this within the last 5 or 10 years. Scientists ask the question for a long time, when you don't need that message anymore, what happens? And we saw them being degraded. We could detect them being removed from the cell, but we didn't know exactly how it was happening until some scientists figured out this process that's called RNA interference. And sometimes it's referred to as RNAi just for short. And RNAi is one way to get rid of those messenger RNAs that are in the cell that aren't needed by the cell anymore. And what happens in RNAi is a couple things. A short strand of RNA is produced, and this short strand of RNA joins this multi-protein complex. And when that short strand of RNA, it's very specific. That short strand of RNA is very specific for the messenger RNA that needs to be dealt with. And that short strand of RNA will join with this complex, and then that short strand of RNA can base pair with the longer messenger RNA. And it'll begin to base pair, and when the complex is behind it, it signals that that messenger RNA, the red strand there, needs to be broken and degraded. And this enzyme complex will then come and it will clip the messenger RNA and break it into 2 pieces. Remember early on we talked about how the messenger RNA was protected down on this end because we put a whole bunch of A's that are modified on this 3' end. So nucleases couldn't come and chew away at this end because those nucleotides block that end from being destroyed. Talked about that several lectures ago. And over on this end we put a guanine that has a methyl group on it, and that blocks that end from being chewed away by some other nucleases that would destroy this messenger RNA. But now, as soon as this message is chopped into 2 pieces with the polyase down here and the guanine here, we've got exposed ends right here and right here that the nucleases can now come in and just chew away from this end, nucleotide by nucleotide by nucleotide, and chew up that RNA back into free nucleotides. And other nucleases can come and chop away this end, and that messenger RNA then can be degraded. As soon as it's split into two, those nucleases have access sites where they can nibble away and chew that RNA and remove it from inside of the cell so that it can't be used anymore. So that's what's going on in RNAi, is that process is happening, and the RNA strand serves as a probe. In other words, it binds to the messenger RNA. You're going to hear that term over and over again, this term probe. We use it a lot when we're talking about biology and molecular biology. It's a bit of DNA that goes in and finds a specific sequence, and here it's finding a very specific sequence in that messenger RNA, binding to it and triggering that messenger RNA should be destroyed and removed from the system. And that way the RNAs are rapidly silenced, they're turned over. Usually those probes are a couple dozen nucleotides, 20 to 30 nucleotides in length, and they're produced by a different system than transcription, but they're made down inside of the cell when they're needed to turn off the use of that particular messenger RNA. Okay, so switching gears just a little bit. Switching gears just a little bit. We've talked about this topic of genomics before. Genomics is basically isolating DNA and sequencing the entire set of chromosomes that organism has. For bacteria, typically that's one chromosome. If you're talking about other organisms like us, it's many multiples of chromosomes. And in humans, you'd have to sequence 2 copies of each chromosome. So there's a lot of genome to sequence and put together in genomics. The first organism that was sequenced was this guy, Haemophilus influenza. The sequence was finished and published in 1995. Shortly, just a few years after that, the human genome was sequenced and finished, and except for we figured out we missed about 10% of the genome about 5-10 years later when we went back and looked at it. So they've wrapped that up again. But Haemophilus influenza was chosen because it's a